Many of you heard Abby and David talk last fall about COVID simulation, COVID in LA County. We're going to hear something completely different this week from Abby. And her title is right up there. You can read for yourself. So without further ado, just go ahead, Abby.
Thank you so much, Duncan. Yeah, so this is going to be a talk on large-scale outbreaks, but large-scale outbreaks of foodborne disease. So quite a different subject matter. And this is a large body of work that I've developed over a number of years that relies on looking at the network structure of the food supply chain and identifying the source of outbreaks in that network structure. So I want to start by acknowledging the number of researchers that I've worked with along the way on this project. So I had a previous postdoc in Germany at the German Institute for Risk Assessment, which is kind of like the German equivalent of the research arm of the FDA. And it's there that I was developing a lot of these methods and applying them to case data from outbreak data from Germany, from recent and historical outbreaks. Okay, so I'd like to start this talk by giving an example of what I mean by a large-scale outbreak of foodborne disease. And so I used to present this talk and say that this was a really large outbreak, 4,000 illnesses, 54 deaths. That sounds like nothing right now in the scheme of COVID, but these are still outbreaks that are preventable, and that we can develop methods to prevent or to really mitigate when they start. And so it's still important to look at these kind of outbreaks. And so this specific example was the 2011 outbreak of E.coli sprouts contaminated with E.coli. And the outbreak was mainly clustered in Germany. There were almost 4,000 illnesses in Germany, but there were cases all through Europe and even imported cases into the US. But most of the outbreak was in Germany, so I'm going to study it in this presentation. I'm going to look at it in Germany. And one thing I really want to note here is that it took investigators 9 weeks to identify the source. And so what that timeline looks like was that the outbreak was identified at the beginning of May, meaning there were enough cases to see that there was, and the strains were sequenced, and so they could see that there was an outbreak happening. But it wasn't until 6 weeks later that the food source of sprouts was identified. And it wasn't until 9 weeks later that the location source in the supply chain was identified. And so we want to reduce this time. And I'll talk about how we propose using network methods to do that. Now, first, I think it's important to get some basic background in the steps of an outbreak investigation, and then we can really show where these kind of network science and data science approaches can help. So the first step is to detect that an outbreak's happening, and then find more cases from the outbreak, and then generate hypotheses about the food item and the location source in the supply chain, and test those hypotheses, solve the source of the outbreak and the point of contamination, and then control by removing the product from the supply chain. And so the work that I'm going to be talking about really focuses on this generating hypotheses step. So how can we help investigators to early on in the outbreak generate hypotheses about what the food item or location source could be, so it can help them to target their investigations. For example, their sequencing analyses, let's target it at these food items or food samples from these locations that we think are higher probability of being the source. And I've kind of been talking about this a bit already, but for generating hypotheses, we have 3 types of that. We have generating hypotheses about the food type source, like was it sprouts or was it meat products, sausage, generating hypotheses about the origin location in the supply chain, and generating hypotheses about the origin location at point of sale. So I'm going to be considering these first 2 types, identifying the food type source and the hypotheses about the food type source and the origin location in the supply chain. The origin location and point of sale, that would be the problem in the case where you have a small outbreak. That's like a few years back we had a number of outbreaks at Chipotle, so it would be identifying the Chipotle that was in question, or identifying that the contaminated food came from a Chipotle. But I'm focused on large scale outbreaks, and so looking at the origin location in the supply chain that could have distributed to all of the contaminated cases that we see in an outbreak. And so just some further background on what the conventional process is to identify the source. So first, there are a number of EPI studies. So food recall, like there are 40-page trawling questionnaires that are given to individuals. And then there are case control studies done with a select group of the patients that have done the food recall study. And then based on the hypotheses that are generated from those EPI studies, triangulation, they do what's called triangulation, which is to collect the sales records from 3 or more of the case patients, collect the sales records from the locations where they think they obtained the contaminated food, and trace them back through the supply chain, hoping to converge on a source somewhere high up in the supply chain. But of course, you can see that there are some limitations to this. It's very resource and time intensive. These sales records have to be collected manually. It's subject to various biases, recall bias. And most importantly, it's often inconclusive in that it returns a large set of frequently consumed food items. So it doesn't do a good job of discriminating to identify the food source. So we want to think about how can data science approaches contribute to this process. And so there have been a few different types of approaches to use data to intervene on this process and to help develop hypotheses earlier on. And the first approach, starting around 2006, was to just use the transactional data that comes from the case patients. So collect all of their transactional data from all of their purchases in the last 2 weeks, because 2 weeks it can be up to like the 2 weeks before their illness onset, because that's incubation period, anywhere between a day and 2 weeks for different foodborne pathogens. And then to look to see, what kind of crossovers there are between different case patients. And so this isn't really data science as much as it's just collecting the data, but still this approach has been quite useful and helped to identify the source in over 20 outbreaks since 2006. Now there's another approach that has proposed using aggregated transactional data from a large number of, from the sales of food products. And so what I'm showing you on the right is the sales pattern, colored by the volume of sales of specific branded products. So each of these are maps of Germany, and they are showing spatially at the zip code level the sales of these different products. And so you can see that there are these different spatial distribution, or rather consumption patterns of different food products. And so the basic idea of this approach that's in this paper here is to compare through a likelihood approach, to compare the relationship between where we see the highest volume of sales of a specific product and the pattern of the outbreak distribution. And so the closer that they are together, the more that you might think that there is a similarity, and that's the likelihood approach. Now, this approach has been applied to one. The original paper was applied to simulated outbreaks, but it was applied to one real outbreak by the Norwegian Food Safety Research Institute, and so it is promising. But it has a few limitations. For example, it requires access to this high volume of sales for products across an entire country, and there are tens of thousands of SKUs of different food products there could be. So if you need that in real time during an outbreak to discriminate between all these different branded food items, that takes a lot of time actually. And so it might not be as readily implementable. And another thing is that it doesn't account for a network structure. And so it's only looking at the last point in the supply chain, the point of sale, but there's all this information in the network structure. And we develop methods that take this network structure into account. And so in the remainder of the talk, I'm going to be talking about the methods that we've developed to take this network structure of the food supply chain and use it to identify sources in the supply chain, be they at the production stage or the processing stage, and to use this network structure for different networks to discriminate between different food item sources. Okay, so there are 3 general steps to our process to using network structure in the food supply chain, and in terms of tracing outbreaks on food supply chains. And the first is modeling the food supply network structure, because this is an incredibly complex system, and we don't have exact data on the supply of all foods to all foods through the supply chain. And so the modeling the food supply network structure, this is one of the main reasons I went to Germany, is to work with researchers that had developed and calibrated a model of the aggregated German food supply system. And so this network model consists of these multiple stages of agriculture, processing, trade, and warehousing, and consumption. And it's aggregated spatially to the I think you can't see it here, but there are 402 counties in Germany, and then 50 import and export countries. And my collaborators used a large number of data sources to create this network structure from production locations. Productions, locations, and volumes came from agriculture and industry statistical data, published statistics. And then import and export information came from customs data. Cross-regional transport flow information came from shipper and shipment survey data. And the zip code level consumption came from retail revenue data. And so taking this together across all of these stages, we have one of these networks for each food type category for 51 different food type categories. And we also have the network of connections between these food types, because you need some of these products to produce other products. And so you have that, this is actually a network of food item networks. And so just a few words on how the model is developed. So the first step is identifying the connections between the commodity categories, essentially identifying these links in this network. And so we might have we have these 51 different food categories, and we need to see how they're linked together. So for example, sugar, milk products, eggs, and grains all are used to create baked goods. And then the second step is to estimate the links between these spatial regions and commodity categories. And just on a very high level, this is done using a gravity model that so gravity models are used to develop to estimate the probability that food goes from one spatial location to another spatial location, each in different stages of the supply chain, from production to processing to distribution to consumption, and across these different food categories. So there are a large number of links that we have to fill in this network, and it's done using a calibration process to transport to German transport data. So every few years there is a major survey that's done on all of the flows of different subcategories of commodities between all pairs of regions or all pairs of counties. And so that was used as calibration data for the model. And so that's all I'm going to say about the network model. But just remember for moving forward that it gives us, for each stage of the supply, it gives us this, the probability or the weight, the volume of food that's traded between region in one stage of the supply to a region in the next stage of the supply in that region and all other regions, for all stages of the supply and all food products. And one thing to note too is that this set of 51 food categories, it's comprehensive. It accounts for all food products. Some of the groups are more aggregate than others. So for example, we have eggs, but then we have another category that is vegetables, which is a much more aggregated product. But we also have vegetables, fresh vegetables, and then vegetable products and vegetable juices. So it's still a little disaggregated in that way. And what commodity categories are used come from the data that was available at the time this was originally developed. Okay, so we have the food supply network model, and now I'm going to talk about the approach for identifying the location source in the supply chain. And so even though in the process flow for investigating a foodborne disease outbreak, you would first identify the food type that caused the outbreak, and then second, identify the location source in the supply chain for that food type. Our method for identifying the location source actually underlies the method for identifying the food type source. And so therefore I'm going to explain that first. Okay, so this is a stylized view of the food supply chain structure that I showed for the map of Germany in a few slides back. And so let's just say it consists of these multiple stages of production, processing, distribution, and retailing, or other types of consumption, restaurants, other points of sale. And so when we have an outbreak, we get reported cases of illness associated with the point of consumption. So these are at first hypothesized, but then they can be ultimately linked to the location where the contaminated food was purchased or consumed. And then our goal is to take this network structure of who's connected to who and the probability that the weight that is transported across these links to identify a probability distribution over all of the possible sources of the outbreak. So in this little stylized example, I'm just looking at this nodes in this first stage of the supply as the feasible sources, but feasible sources can be any node in the network that can connect to all three of these nodes. And so our goal is to develop this probability distribution over the set of feasible sources. And we can also develop a maximum likelihood estimator for the source location, which essentially just looks at the highest probability node in the highest probability in this distribution. So this problem of identifying the source of an outbreak in a network has been studied by a number of researchers in the last decade or so. But in this paper that I have cited down here, my colleague and I review these cases and describe why they don't apply well to these approaches, and why these approaches don't apply well to the case of foodborne disease. And foodborne disease, and identifying the location source of an outbreak of foodborne disease in the food supply chain network requires a different approach. And so the main reason is that this is not a contagion process. This is not an SIR process. We actually model food distribution as a diffusion process. And so we can think of each food item as being contaminated at its either in its production or processing. Those are the two really likely places that the food will get contaminated: the production or the processing stage. And then the food, you can think of it as traveling in different units, like containers, through the supply chain, and it really the contamination doesn't spread that much once the food is transported through the supply chain. There can be some small amount of growth in the contamination, but really what you'd see cross-contamination between food products, or commingling of contaminated and non-contaminated items, but you don't see this contagion process happening. And that's partly because you have a lot of packaged food items, but it's also partly based on the way that foodborne contamination like E. coli or Listeria or Salmonella will grow on the food item, or even within a package. And so so that's one of the main features that we have to design for. It's not a contagion process. And then another feature is that food can be distributed across a large number of possible paths. And so what I'm showing you here is an example from the EHEC outbreak, the 2011 EHEC outbreak, where I've circled in red at the top here the location source, which is eventually traced back to this small organic farm in northern Germany. And down here, I'm circling a large cluster of cases in different restaurants in Frankfurt. And on the right, I'm showing you a number of paths that the food could have taken to travel from Hamburg to, or not Hamburg, sorry, but the source in Olsen to these restaurant clusters in Frankfurt. And you can see, based on the weight of the links, that these paths, a lot of them have very similar probabilities. And so the reason I point all of this out is because a lot of the methods that had already been developed were based on shortest path assumptions or highest probability paths assumptions. And so they made those simplifications because in the cases that they were applying their methods to, that could be a way to discriminate between to all of the different possible pathways that could be traveled across in a network. But in our case, we can't do that because we have a large number of paths of similar probability and almost the same length, the same number of stages of network steps to take. So we account for these multiple stages of supply through a random walk model of contamination transmission. So we assume that the probabilities of going from one node to another node are Markov probabilities. And we can define, taking that together across the whole network, we can define a Markov transition matrix to represent the network. And this Markov transition matrix represents the transitions between transient nodes to transient nodes and transient nodes to absorbing nodes. And so to make it clear, the absorbing nodes are the consumption nodes. So it's where the food is purchased and consumed and leaves and gets absorbed, never to re-enter the flow through the Markov chain again. And so we can see this essentially as a observing Markov chain, the flow of food through the food supply chain. And I should also note that the way that we've defined our Markov transition probabilities is the relative food flux out from a node. So that we have the weight of food that's transported from one node to all of nodes that it's connected to, and we just take the relative weight to get the Markov probability. Okay, and then excuse me, we can estimate the source location using Bayesian inference if we have a prior distribution over the sources. So our goal is to determine the probability that any feasible source, S and omega, is the true source, S star, given the observation set theta of illnesses. And so we'll be coming back to this observation set theta a lot through the rest of this presentation. And so we can define this probability, the Bayesian, the probability of being the source given this observation set as just Bayes' laws here, the prior probability times the likelihood, which is the equal to the probability of the observation set given theta over some constant. And if we want to identify the maximum probability source, we can do that through a maximum probability estimator as well. And so importantly here, our likelihood is informed by the random walk transmission model that we assume is how food, that I showed in the previous slide, and is what how we assume that food is transported across the food supply network. And so this results when we use Markov chain probabilities, this results in a probability mass function, a PMF, over all the feasible sources. And if we want the maximum a posteriori probability estimator. And I'm going to point out a couple features of this. So that this probability that we're looking at here in the PMF, or the estimator, incorporates the probability of traveling through the network from any source to any observation using the Markov chain absorbing probabilities, so that we can account for the probability of going from S to from the feasible source node S to the observed contaminated node O across any possible path. And then we assume that these individual observed cases are independent. And with that assumption of independence, we can just take the product for all nodes in our observation set of their probability of going from the source to that observation node. And so this approach, with this independent assumption, allows us to look at this probabilistically, and it allows us to account for a large number of for all the paths of travel that could have been taken through the network. Okay. And we can apply this to, so I'm going to actually just show results from applying this to the 2011 outbreak of E. coli and contaminated sprouts. But of course, we've done a large number of accuracy tests, and if you're interested, you can look at the paper. But essentially, we see that the accuracy of the approach is higher than approaches that were not developed for the foodborne disease situation. So for example, approaches that make a maximum, that look at only the highest probability path that the contamination could have traveled through the network, of course, isn't going to do as well as the approach that looks at all of the paths that could have been traveled. Okay, and when we apply this to the 2011 outbreak of E. coli and contaminated sprouts, we are able to identify the source location as a county, as one of the German counties. So what I'm showing you here on the left is the map of Germany, where the red dots indicate groups of 10 infections in each of these counties. And the shading represents the PMF, the probability mass function over the sources that we got from applying our traceback method. And so this is where the source location was in the supply chain. And based on data as early as week 2 of the outbreak, we were able to identify this as the most likely source in the supply chain, or yeah, the most likely county level source in the supply chain. And I'm showing you on the right the 2-dimensional, just the bar probability distribution in the same of the same PMF. Okay, so that's our method for identifying the location source. Now we can use that to identify the food type source of the outbreak. And so the question that we ask here is, given these different network models, the way that we frame the question of identifying the food type source is to say, given different network models of the food supply for various food products, how do we identify the most likely food type source network, which I'm going to call N star. And so here I'm showing you different networks of distribution for different food products. And the question is to determine which of these food products can best support the observed outbreak distribution. And so just remember this from one slide back. This was applying the source location estimator to identify the source in the supply chain. Well, what I didn't make clear was that I had assumed that I had used the vegetables network to do this. And so we need to know what we need to identify this vegetables network first before we can apply this location estimator. And so that's the goal of this approach now, is to identify that this vegetables network was the true source. And we do that by looking at the information in the distribution of this PMF, and I'll talk more about that. But just for the following examples, just remember that we have this set of 51 different networked food type commodities to work with, food type categories to work with, and for each of these we have a different network structure. And in the following, I'm going to be just illustrating the method on 6 different food types or food type networks, which represent which were a number of which were hypothesized sources during the EHEC outbreak, which is partly why we include them. And then the others are just frequently very highly consumed food products in Germany that also have had a history of foodborne disease contamination or foodborne contamination. And so the networks are cheese, eggs, milk products, which incorporate a number of dairy products, meat products, poultry products, and vegetables. Okay, and so the basic idea of our approach is to look at the information in the PMF that comes from tracing back outbreaks on networks. And I'm showing you an example here, which shows I've simulated an outbreak on the vegetables network. And on the left, I've traced that outbreak back on the vegetables network. But on the right, I've traced back that outbreak that was simulated on the vegetables network on the egg network. This is incorrect. This should say simulated outbreak on the vegetables network. And so what we see is that there's a lot of structure in the PMF that results from tracing back the outbreak on the network it was simulated on, versus almost a uniform distribution on the when the outbreak that was simulated on a different network is traced back on a new network. And we can see that a little bit more clearly when we rank order the PMFs from highest to lowest probability mass. And so we can see pretty clearly from this that there's a lot more structure or information in the PMF that results from the food network and outbreak distribution that was a true match. And so we can say that this is a strong signal of agreement between the network and the outbreak distribution. And so this is essentially the essence of our method, for identifying the food type source of an outbreak. Now I'm going to get into a little bit more of the detail of how we do it. So we refer to the probability mass function over the feasible sources, S in omega, that's the set of feasible sources. We will refer to this PMF derived from tracing back an outbreak distribution or an observation set, as I was calling it earlier, theta of size C cases on the network N and I. We will call it chi. So chi N I theta C represents the probability distribution from tracing back theta, the outbreak distribution theta C on network NI. And this is a PMF over all sources in the set of feasible sources omega. And then I'm also going to define, we have 3 different flavors of outbreaks and their associated PMFs. So first we have what I call the real outbreaks. So this is the real outbreak being investigated in an individual case. And so, for example, this could be the 2011 EHEC outbreak when we were investigating that. That was a real outbreak. And I'm showing you also its associated PMF that came from tracing back that real outbreak on network NI. In this case, NI is the vegetables network. And here I'm showing a simulated outbreak. So it's another outbreak that we just simulated on the vegetables network. And I'm also showing it, the red dots again represent groups of 10 cases of illness. And you can see that there's like a clustering of cases in certain location that came from how the outbreak was simulated. And this gives us an associated PMF from simulated outbreaks. And then finally, we have these, what I call randomly sampled outbreaks. They're not really outbreaks, they're random sampled of random samplings of the consumer nodes in the supply chain, which represent the consumption at the 402 different counties in Germany. And I randomly sample quote unquote illness cases at these counties in proportion to the population that live in each county. And so I'll call these randomly sampled outbreaks, even though they're not really outbreaks. They don't result from a source giving rise to it as an outbreak. It's just a sample of essentially of individuals or of individual counties. And this gives rise, we can still trace back this random sample of cases. We can still trace this back, and we can get an associated PMF from tracing back this random sample. And so then I'm also going to talk about, if we want to measure the amount of information in a distribution, what are the right metrics to do that? So so the first metric probably that comes to mind for measuring the amount of information in a single distribution is entropy, the information entropy, which this is, it by definition measures the amount of information in a single distribution. So on the right, I'm showing you a uniform distribution and on a uniform PMF, and on the left, I'm showing a PMF that has one, if you can think of these as being ordered by the source ID, one source with a very high probability and the rest of the sources with very low probability. And so this PMF on the left has a lot more information, and it has a much lower entropy than the uniform PMF. And so the entropy measures the amount of essentially the less structure in the distribution, the higher the entropy, because it's the less there's less information in the distribution. And we can also talk about the inverse of the entropy. So if we want a measure of if we want kind of a measure of the structure in a distribution, that the more structure that we have in the distribution, the higher the measure, then we can take the inverse of the entropy so that we get this distribution on the left with more structure having a higher value of this metric than this on the right. And so with that in mind, we define what we call the baseline characteristic signal for each network, which measures the amount of signal that occurs between a network structure Ni and an outbreak distribution theta of size C. And we define this as the inverse of the entropy, again, because we want to have an increase in this measure of baseline signal be proportional to an increase in the amount of signal between a network and an outbreak distribution. And we can get these baseline signals, these baseline characteristic signals, from simulating outbreaks and tracing them back, or from taking what I call these random sampled outbreaks and tracing them back. Okay, and so what do we find when we apply this metric to simulated and random sampled outbreaks on each of our 6 networks in the set of networks that we're considering? So on the left, I'm showing you the results from simulating and tracing back 1,000 different outbreaks on each network and each of these 6 networks. And I'm showing you the mean and the 95% confidence interval from the simulation of at each number of illnesses. So I do this at each number of illnesses. I recalculate this at the PMF at each number of illnesses, because as you get more information and more illnesses, the PMF will change. So what we see here is quite interesting. There is a clear difference. There are clear distinctions between the networks in their baseline signals from simulated outbreaks. And so some networks have, on average, much higher baseline signal than other networks. On the right, I'm showing you the PMFs, this measure of the baseline signal with the inverse of the entropy of the PMFs that come from tracing back these random sampled outbreaks on each network. And on this is an inset that shows a zoom in. This is on the same scale as here. So we can see that the simulated outbreaks have higher baseline signals than the random sampled outbreaks, as we would expect. But we still see, when we zoom in, we still see a lot of structure in the random sampled outbreaks. And the structure, the ordering of these networks is different between the random sampled outbreaks and the simulated outbreaks. So what this tells us is that there are these consistent differences between networks and their baseline signals, both from the upper bounding simulated outbreaks and the kind of lower bounding randomly sampled outbreaks. And this is something, this means that we're not able to just compare an outbreak, a PMF from one network to the PMF from another network. We're not able to compare those directly because we can see that some of these networks have a higher characteristic signal than other networks. So we need a control for this structure in developing our measure of the signal, what I call the signal resonance between a network and an outbreak distribution. And so this is just what I just said, that we need a control for both that upper bounding, that high structure of PMFs that come from simulated outbreaks, and the lower bounding structure that comes from PMFs from randomly sampled outbreaks. And so if we're talking about measuring the amount of information between two distributions, we can use the relative entropy, which is the Kullback-Leibler divergence. And this measures the information gain between two distributions. And I'm going to call this the DKL, the divergence, the Kullback-Leibler divergence. And it's defined similarly to the entropy, but it takes the log of the ratio of the value of one distribution over the value of the other distribution. And so we can use this to define, now I'm kind of bringing everything to get together to define this normalized signal resonance measure. And so to do this, we have three different pieces. We have, first, I call the simulated signal relative to random. This is the Kullback-Leibler divergence between the simulated, the PMF that comes from tracing back a simulated outbreak on a network, and the PMF that comes from tracing back a random outbreak on a network. And so this is actually independent of any real outbreak under investigation. This can be calculated in advance for any given network because it's a measure of the difference, the divergence between simulated and random outbreaks and PMFs on that network. And then we define what we call the real signal relative to random for a network and an outbreak distribution. And so this real signal is the Kullback-Leibler, is equal to the Kullback-Leibler divergence between the PMF coming from the real outbreak and the PMF coming from the random outbreak, both traced back on network NI. And so we get these two pieces, and then we just define the normalized signal resonance measure, which I might call Norm-Sig, as just the ratio of the real signal over the simulated signal relative to random. And I just wanted to note that that we don't look at the Kullback-Leibler divergence between the real PMF and the PMF from the real outbreak and the PMF from a simulated outbreak. And the reason for that is because this Kullback-Leibler divergence is a positive metric, and it can't tell us the which distribution has more information than the other. So we compare both the simulated and the real outbreak to the random outbreak because we know that the simulated or the real will have more structure than the random. And so this is our final result. This is our final metric, the normalized signal resonance metric or measure. And here is a kind of an overview of how the whole measure works together. I think I'm going to skip over this because I want to show a result, but I can come back to this if people have questions about how it all fits together. Okay, so what I'm showing you here are accuracy results after simulating a large number of outbreaks on each network. And on the leftmost graph, I'm showing the measure of the normalized signal, the Norm-Sig measure itself for the true source network. And so we can see that this converges to 1 over a large number of simulated outbreaks, which is what we expect, since essentially when we're doing this over a large number of simulated outbreaks, we're comparing the real signal is the same as a simulated signal, because in this case we're looking at simulated outbreaks. So these real signals are really simulated, the simulated signal relative to random. So this converges to 1, as we would expect. And then I'm showing the rank and the accuracy of the results over a large number of simulated outbreaks. And so on average across all networks, we get an accuracy of around 80%, being able to identify the true source network in the first ranked position over the ordered rankings of all the networks. But the accuracy for individual networks, some are much higher than other networks. So that's something that we might want to look into in the future, is what causes that difference. Why is it easier to trace back and to identify the network in certain network settings than other networks? Okay, now I'm going to show applying this network, this normalized signal resonance measure to the 2011 outbreak of E. coli in contaminated sprouts. So we know, I showed from earlier, that sprouts were identified as the food item source of the outbreak around week 6 of the outbreak. It was earlier on that hypothesized food sources came out of some of the epidemiological studies, and these were mainly vegetables found in salad, yogurt and other dairy products, and meat products, especially sausage. So we can investigate, we can compare between these different food products, and also include some of these other products that are, like I mentioned at the beginning, are highly consumed food products in Germany, weren't necessarily hypothesized as sources of the outbreak, but still are helpful to use in our investigation set, because null results are also useful. So if we see no signal for a network, that may be just as useful as where we see a signal for a network. And so indeed, for this outbreak, as of the number of illnesses after the first 1 to 200 illnesses, we were able to identify in the first 2 ranked positions the vegetables network, which is the network within which the sprouts were contaminated, as the most likely food item source. Now, we identified the meat products network with a slightly higher normalized signal measure than the vegetables network. But in a case like this, we would definitely give results like this to an investigator and say that this would indicate that these 2 networks are the most likely hypothesized sources, and that we essentially see null results for these other kind of food type networks. Okay, so I'm going to just summarize. I have shown that using these aggregated food type category networks can help investigators to identify and discriminate between hypotheses for the food item source early in the outbreak and help to focus the investigation. And that this network model that we're using is aggregated at this spatial county level, and it comes from estimated gravity model links between all of these different parts of this supply structure. But despite these large number of estimations that went into its creation, case studies suggest that kind of modeling can provide meaningful information for distinguishing between these higher level food type sources of an outbreak, or the aggregated location source of an outbreak. Food networks are structurally unique, and we saw that it's important to consider the unique structure of networks in the source identification problem. I think that one thing to say is that more science is needed to identify the relationship between network structural features and the characteristic signals and the accuracy of source identification. And we started looking into this with a large number of different network features, and see that there is a correlative relationship with the accuracy of traceback. And so that might be, or the accuracy of being able to identify the network as the source. And so that might be a way to help identify when we can and cannot get high accuracy for a network. All right, so this has been a team effort, and I'm acknowledging my team and the funding sources that went into this project along the years. And I'm open for questions in our remaining 5 minutes or so.
Well, thanks very much, Abby. That was a fantastic talk, and really interesting. Questions?
Abigail, great talk. This is David.
Thank you.
So just pursuing sort of your last slide and ideas around that, because, you know, this is focused on saying here we have a given observed pattern of a real outbreak. Can we identify the network? But, you know, one of the things you talked about was sort of the ratio of a simulated food network to the random. And then each one of these food networks has characteristics. Could you then use that information and the characteristics to say, well, what's the outbreak potential in a food network? And then across all the outbreak potentials, what are the characteristics that are common that maybe could be used from a public health point of view to say, well, let's optimize the food network so they avoid outbreak potentials? Is there something, am I thinking along a line? I mean, that's kind of what I was thinking with your last slide there, not the characteristics for identification, but the characteristics for public health intervention in food network outbreaks.
Yeah, that's really, really insightful. I actually have another paper, which I didn't mention, where we look at different network structural features and look at, I think we looked at the heterogeneity of the degree distribution, like how much, essentially, like, are all nodes connected to, are there some nodes that are connected to a very large number of nodes, and then a lot of other nodes that are not, are very low connected.
Mm-hmm.
And we looked at measures of centrality as well. And we were able to develop a way to characterize the amount of the probability that we would be able to trace back outbreaks in that network. And so we suggested that that could be used to inform where we would want to put more resources into identifying the structure of, into better mapping that supply network structure, so that when there is an outbreak, it's easier for us to trace it back, because with the existing model, it was very low accuracy. But I think even what you're suggesting is taking one step back and saying, what can we expect about the distribution pattern and the virulence of an outbreak, like how well distributed you can see an outbreak being. And that kind of, if we can target interventions there, that kind of nips the problem in the bud before it even gets to the outbreak. And that's something I haven't looked at as much. And there's definitely a lot of potential there if you have a good network model. Thanks.
Thanks.
Partway through, you showed us a 2D plot, well, a histogram of posterior probabilities of sources, and it showed your maximum likelihood estimator over on the left of that plot, but a whole bunch of others right nearby that I assume were sort of geographically similar. And then there was another peak way over on the right-hand end, which was probably someplace quite different. I would think that public health officers starting an investigation would like to know not just the MLE, but that posterior distribution, and maybe be investigating a number of different sources. Is that right?
Absolutely. And it almost feels pointless to even bring up the MLE, because that's not, these approaches are very estimated, and so they can indicate a set of plausible, more higher likelihood hypotheses than other locations or other network sources, but they definitely can't be used to say definitively this is the network that is, or this is the node that is the true source. So really, we're always considering the top ranked set, or like you you pointed that out really well, which is like you see these kind of groups of high probability nodes, and you even saw that in the spatial probability distribution over the sources. You see that there are areas that have high probability and areas with low probability, groups of counties. And so I'm kind of in the paper that we had explaining the traceback method, the source location method, we call it a maximum likelihood estimator, but it's really not. It's really about identifying that probability distribution and the set of likely sources. So,
do you recall whether that cluster over on the right of that figure was geographically far away from the one on the left?
It is. I think if I can go back, I think we can even see the most recent time I see it here. We can see that there's one cluster around here and one cluster around here. So they're pretty far. They're pretty far in the scheme of things. They're definitely not linked. And then you can even see them in different parts. So like if there's a location that is producing a large amount of a certain product, or there are multiple locations that are producing, in this case, vegetables, then you could see those as both like two very different locations being highly likely identified sources.
Mm-hmm. Other questions?
I have a question. It was really interesting, the talk. Thanks a lot. Have you tried this method on other outbreaks after the E. coli outbreak?
So that's a very good question. We suffer from a lack of real examples. So I don't have another set of real outbreaks to apply this to. And I think that's a real limitation with and whenever we present to investigators, it's a real limitation in their ability to have confidence in this method and being able to use it. I think it's good that we don't have a large set of outbreaks that we can apply this to. But at the same time, there have been outbreaks and we just haven't traced them back to a source. So I have no way. I can apply the method and I can say, oh, it looks like it was the meat products network, but I have no way to establish that. And that, unfortunately, that's normally the case, is that the source isn't identified. The pathogen is identified. Maybe the food type is identified, but the location source isn't, or something like that. But
it does seem like collecting all this data and doing the analysis is a labor-intensive process. If you were faced with a new outbreak, would you be willing to go through it again? And how much effort did it take now that you've got the methodology established?
Yeah. Also a very good question. So it would take a while to develop the network model that we had for Germany and the US if I wanted to look at outbreaks here. So that would be a large initial investment step, which I would be very interested in developing that network model of the US food supply chain for application to many different problems. I think that relates to a lot of the work that I do with Kayla in terms of looking at and Bernardo, looking at the food supply chain and looking at how the way that supply chain set up can impact what food is accessible to people in various locations. And so I think there are a large number of applications of that network model. If we had that, then I would be ready to apply outbreaks as they come in. And I think I'd be very willing to do that, but I need that like big initial investment project to develop that network model of the US.
Other questions? Well, if not, let's give her a virtual handshake. Thanks very much for another really, really interesting talk.
Oh, thank you so much. Yeah, to be honest, I was a little I was a little apprehensive about talking about this work because it's so different from the COVID modeling work and the other work that we do. And it's not really traditionally biostatistical work, but I hope that, you know, I was able to explain it in a way that was able to register, and that was able to start to see some relationships between that and some of the other work that I do here. So.
Well, thank you. And I'm glad you did decide to do that.
Thank you. Thank you so much.
Okay. Bye-bye, everybody.
Okay.
Thanks,
Bye.
Abigail.
Thank you. Thank you for attending.